Okay, welcome back for the last lecture in 1442. I am super sorry that I can't be there in person, but I didn't want to miss the material for this one, so I'm going to go ahead and record the lecture material for you. And I just want to make one note before I launch into the content here. And that is that the last section of notes, so this is back on Canvas, the PowerPoint 22, Ecosystems and Conservation Ecology, I updated that today. And it really just updated the conservation section, the stuff I'm going to cover now. And it was mainly to update a couple of the graphs and make the slides prettier. So nothing major, but just wanted to let you know that if the PowerPoint that you downloaded for lecture on Tuesday, there are going to be a few slides that don't match with what I'm going to show in this video. Okay. So we got through learning objectives 1 through 6. That was mainly the ecosystem ecology part of module 14. And then today I'm going to cover 7 through 13, which has more to do with the conservation biology, conservation ecology aspect of this module. Okay, so just a little bit of review. You've already seen this when we talked about community ecology, but when we start to think about conservation, we are thinking about the biodiversity of a system. And we talked about in community ecology how if, for example, you were walking through this forest, these two might have a very different feel because even though they have the same number of species, the distribution of species within those ecosystems or those communities is not the same. A main focus for conservation biology is going to be the species richness, whereas we were a little more focused on relative abundance in terms of the community diversity. So remember, species richness is just the total number of different species in a community. If we look at measuring biodiversity in that respect, sort of species richness, it really includes all of these things. So the genetic diversity, if we were to go in and look at the different species of spiders, for example, or trees, and if you think back all the way to our evolution part of the course, and we talked about inbreeding and things like that, when populations get small, you get more homozygosity, the genetic diversity goes down. Genetic diversity increases the likelihood of extinction because you're getting more homozygous recessives that may cause disease, things like that. Hard things to recover from genetically when you get small population size. So genetics is an aspect of biodiversity. Also, species diversity. We've been talking about that recently. And then you can also think about it in terms of ecosystem diversity. And we also showed graphs like this for the community ecology, where around the tropics you tend to get more diverse ecosystems. And in the context of ecosystems, where we're talking about nutrient cycling and abiotic-biotic interactions, the most productive ecosystems tend to be around the equator as well. So all those three are aspects of biodiversity. One of the key observations in terms of where biodiversity or how we maintain biodiversity is that there's a relationship between the area that species inhabit and the area and the number of species that can inhabit that area. So, for example, I've got two graphs here showing birds and beetles. They show this same positive trend where, right, not so surprising, when you have more area, you have more species of birds. And I say maybe that's not too surprising, but you could potentially just see more birds of the same species, but that's not what we actually see in nature. We actually see that if you have more area, you also tend to get more species. And that's true across a wide variety of organisms. So species richness increases with the area. That is the species area relationship in a nutshell. Um, when we start to think about conservation biology, that species area relationship, there are a couple other factors that we need to consider when we start to think about, okay, how many species do we actually expect to see there? And that is, how isolated are the areas? Because that's going to affect species richness, which where we're headed is to island biogeography. But before we get there, it's important to realize that what you think of as an island, this uh, sort of body of land in the middle of the water, is not um, sort of what all islands are. So this little parenthetical I have up here, not all islands are islands. You can think of mountains as islands. So, right, I think these are the Tetons, uh, where you get similar relationships. And here, this is showing montane mammals. So same kind of relationship that we saw with the birds and the beetles on this previous slide, where the larger the area in the mountain habitat, the more mammal species you find. Similarly, you can think of lakes as islands. This is almost the opposite of what you typically think of as an island. But these lakes, and this is in northern Wisconsin, just this little graphic, right? These blue areas are, in effect, islands of aquatic habitat. And you can see that as the lake size increases, the number of fish species in those lakes tends to increase. So those kinds of observations led to this theory of island biogeography. So this is learning objective number 8. And the question was, what mechanisms might increase the richness on large islands, or conversely, reduce the richness on small islands? What explains this relationship? Why don't you just see more individuals of the same species? The theory is mostly attributed to a couple publications by these guys, MacArthur and Wilson. This is the E.O. Wilson that we talked about on Tuesday, and we're going to talk about again in a few minutes. And they propose this equilibrium model where there's a balance between the rate of immigration and the rate of extinction. So I'm giving you just the definitions for those terms. The rate of immigration is just the arrival of new species on an island, and the rate of extinction, the rate that they are going extinct on that island. This theory of island biogeography does a pretty good job of predicting the number of species that you're going to find. So, in other words, explaining the species area relationships that we see. I'm hoping you can see my cursor, but I'm not sure. Just going to leave it on visible. Maybe that works, just in case. So, what is the equilibrium model of island biogeography? Well, first of all, these two lines, the blue one is showing the immigration rate of new species, and the red is the extinction rate of new species, extinction rate of species that are on the island. And where those two lines intersect is going to be the number of species that you predict to be present. So that is the equilibrium number of species for a given rate of immigration and extinction. So you can imagine immigration has this sort of downward trajectory, because when there are very few species, all the new species that arrive on an island are new, or so if you think of it in the extreme where you've got 0 species on the island, everything that arrives is new. As you get more and more species, fewer and fewer of them are new. And so the rate of immigration of new species is less and less. For extinction, the rate increases with the number of species for a couple reasons. One, the pool of potential species to go extinct is bigger. But also, as population sizes increase for all those species, the number of individuals per species must go down. And so that's going to also increase the rate of extinction uh, per species. And then as more species get on the island, as the island gets more populated, the potential for competitive exclusion increases. So going back to the sort of population ecology lecture where they're excluding each other from certain habitats. And like I said, the equilibrium is going to occur when those two lines intersect or where they intersect. So how does the island area affect this? So here we can see the immigration rate for a nearby island. So this is an island that is near to, if you think of it as islands in the ocean, for example, it's near to the mainland. But you could also think of it as lakes that are nearby to other bigger lakes, just as an example. If you're in a nearby island, the rate of immigration is going to be higher because it's sort of less effort for a species to move from mainland to island. As islands get farther and farther away, that immigration rate is expected to be lower. We also talked about this again way back in the beginning when we talked about Darwin's insights from biogeography. So hopefully some of this maybe rings a bell. The flip side, the extinction rates, if you're on small islands, right, you're going to get more crowded more quickly. And so extinction rates are expected to be higher on small islands and lower on large islands. So you can see, like, if you have those sort of four disparate near versus far, small versus large, any combination of those is going to give you a different expectation for the equilibrium number of species. So, for example, if you are on a faraway island that is small, so you're on the low immigration line and you're on the high extinction line, then you expect to have very few species. On the other end of the spectrum, if you are on a nearby island, you're on the high immigration line because you're nearby, and if you're on a large island, you have a low extinction rate. You expect to get lots of species on those kinds of islands that are near and large. Okay. So this essentially is the theory that explains and can predict pretty well if you can get measures of island size and immigration rates and extinction rates, it will predict pretty well how many species you expect to see for a given island size. One additional point here is that the species composition isn't necessarily static. It's one of the cool parts about this model is that if you go back and sample the islands over time, you don't necessarily see exactly the same species, but you see the same number of species. And so we call this species turnover, and the model still fits pretty well. Okay, so conservation biology, moving on to learning objectives 10 and 11. Again, this is the same E.O. Wilson. He has been around and contributing for a long time. His acronym for the major causes of biodiversity loss globally is this HIPPO acronym. So habitat destruction, invasive species, pollution, population growth, mostly talking about human population growth, right? We're growing so fast. And then overharvesting. Uh, the current background extinction rate, I think this is also in the sixth extinction book that you're reading, is about 1 million species per year. Somewhere on that order of magnitude, which that's a scary high number. I've linked here a talk. It's pretty long, so I'm not going to go ahead and play it for you now. But where he goes through Hippo and some of the examples for why these are important concerns when we start to think about conservation biology. This is also a good point to a good spot to point out the connections between what we've been talking about with conservation, or what we're going to talk about with conservation biology, and these different levels of study in ecology. So habitat destruction tends to be an ecosystem level concern. So ecosystem ecologists study habitat destruction, whereas invasive species tend to be a community ecologist level of study. Pollution, population growth, and overharvesting are all population ecology level questions for the most part. And then one that doesn't really fit so well is that global warming, climate change. I suppose um, it's happening at the biosphere level, but uh, ecosystem ecologists, maybe I should write ecosystem ecology there. Um, but just highlighting how all the different levels of um, researchers, whether they're working at the ecosystem, community, population ecology levels, all tend to focus on conservation biology to some extent because it's such a major concern nowadays. It really started to gain traction back in the 60s with the publication of this book, Silent Spring by Rachel Carson. And the book, one of the main things it focuses on is the accumulation of DDT in the environment and how these toxins are accumulating. And essentially, I mean, her focus was on birds, uh, where this accumulation kills the birds. And one of the things that came out of that, and there's been a ton of research on it since then, is this idea of biological magnification, and sometimes called bioaccumulation. So some toxins, like DDT, has a long half-life in the environment. And I mentioned this on Tuesday. I'll show it again on the next slide. But as you move up the food chain, these toxins accumulate in at the different trophic levels. And so by the time, even if it's harmless in the sort of aquatic habitat, for example, by the time you get to the top level predators in that system, it's toxic at a meaningful level, has bad physiological consequences. Uh, PCBs in the ocean. So we've heard lots about plastics and also having harmful consequences in marine habitats. So this and some more links if you're interested to go look at the research on that. Um, Couple more slides just showing that biological magnification. This is one with heavy metals, mercury in particular, runoff from mines, coal plants, volcanic activity. Even if the level in the water is low enough to be safe, so if you're really low down on the food chain, like primary consumers, krill, maybe secondary salmon, pollock, oyster, you, the bottom here is showing the recommendations for how much you can eat of the, like humans could eat of these and still be within safe limits. Basically, not limited on these. Not that we ever eat krill. I don't think krill is a thing for people, but salmon, pollock, oyster. You can eat those, and they have a low enough level of heavy metals that you're probably safe. Now, if you're eating trout or tuna, which are maybe tertiary level consumers, limiting that, the advice is limit that to a few times a week. And then if you get to the top level predators in any of these aquatic systems, you should probably not eat that so often, right? A few times a month at most, in order to keep the amount of heavy metals that you're ingesting at a safe level. Okay, and what we mean by safe, I mean these are, everybody has a different risk tolerance. Okay, and everybody's metabolism is different, but these are the sort of FDA uh, and EPA uh, recommendations for consumption. PCBs, not just marine environments. That's why I left this graph in. Um, they accumulate in freshwater habitats as well, right? Zebra mussels, these are like freshwater clams. And then as you go up the food chain to these top level fish, eating walleye, you're going to eat a not insignificant amount of PCBs. Probably shouldn't be eating that every day. When we think about biodiversity in terms of conservation, there are a few ways that it's useful to prioritize where research and conservation efforts need to go. The most widely recognized sort of international system is the IUCN, and they produce what's called the Red List. Uh, it has seven levels or seven categories and here they're listed from the least concern, where they're widespread and abundant, to things that are extinct as far as we know. There are no known individuals. The three that get the most attention are here in the middle because these are the ones that are threatened. So they're still around, but they're in trouble. So you can see vulnerable, endangered, and critically endangered in increasing levels of risk, and that tends to correlate strongly with the number of individuals that are still wild in the habitat. A couple other, just I point these out because it's going to show up on a graphic later. There are a bunch of species that are data deficient, and that shows up often as well, where we just don't know enough to put them in a sort of not much concern versus a threatened species. And just want to highlight here, because we're going to talk about these in the next few slides, the conservation strategies for things that end up in these three middle levels of threatened. We focus on resource management, movement corridors. I'll talk more about why those are important when we get to that slide. And then habitat restoration. What can we do to restore the habitat? Because in many cases, right, going back to the hippo thing, there's not one cause for why a species is endangered. Is it toxins in the environment? Is it habitat loss? Is it human encroachment? All of these things may be factors. So there's not just one mechanism there's not one bullet to solve all the trouble spots. One of the examples for resource management I think is maybe the easiest to think about is the case of overharvesting, right? So overharvesting isn't a case where it's habitat loss that is causing a species to be in trouble, threatened. It's that the harvesting of that organism, and fish are the easy one to think of. So all these pictures are about fish. Um, it's harvesting that renewable resource, but at a rate that's not sustainable. So, for example, fish are going to breed, they're going to produce more of them. That's a renewable resource in some sense. But if you are fishing them on a commercial level that takes them out of the habitat faster than they can reproduce, that's not sustainable. And this goes back to when we think about, how do we employ research management strategies, we run into what we talked about, I forget if it was Tuesday or last Thursday, this tragedy of the commons, right? Where multiple groups are competing for the same resource. In this case, they're competing for the fish. So everybody is sort of acting in their own self-interest to get the most fish. And if there was only one person doing it, it wouldn't probably sort of outpace the renewability of the resource. But because there are many people competing to do it better and better to get their own fish, it leads to that overexploitation. And so the solution is resource management. The problem with the resource management is that it's not always easy to enforce. So you can impose catch limits, you can put seasonal restrictions on it, you can ban certain types of harvesting, so certain types of netting or trapping, and you can implement protected areas where people just can't fish. So there are nurseries where fish go to breed. So if you um, sort of make those off limits to fishing, it at least gives those fish a better chance to be sustainable. Uh, the other two conservation strategies tend to fall into this category of restoration ecology. And these do have more to do with habitat destruction being a main source of the vulnerability of the species. And this is where applied ecology turns to sort of help and why sort of basic ecology is important so that we know how to restore degraded ecosystems into what would otherwise be functioning ecosystems. So, movement corridors are something that has gained a lot of traction, and the way to think about these is they connect islands. So, there are a lot of species that this four-lane highway would essentially be an insurmountable barrier. So, you've got an island of forest over here on the left and an island of forest here over on the right. If you can connect those, think about the island biogeography principles, where instead of having two smaller islands, you now have one bigger island. So the more you can connect the islands, the bigger the area is, the more species you can maintain. That is what corridors are all about. I have a lot of interesting personal experience with this, doing ecology in Florida, where every spring there are these massive amphibian uh, breeding migrations, where they're all going to the ponds to breed, and they are crossing roads like crazy. To the extent that it's dangerous at times, like the roads literally get slick with things that have been run over. That's kind of gross to think about, but it happens. And so one of the strategies, both of my pictures here are showing bridges that are meant to be corridors, but piping, culverts that go underneath the highways are also a strategy, especially in places where amphibians are a main concern for the migrations. But at the end of the day, it's all about connecting the habitats so that you can maintain higher species diversity. Another example, Chinook salmon, another a couple of examples with videos here where taking the dams out and doing coastal rehabilitation has led to a resurgence in the Chinook. Uh, so basically just putting back the coastal habitats and the rivers the way they would have been sort of over evolutionary time for these things has um, done a lot for the Chinook populations. And then last example, reforestation is a big one. This is, basically planting trees so you restore the habitat. This one's gets a lot of attention and has done a lot of good because it both restores habitat that sustains lots of species, but it also combats climate change because um, they take carbon dioxide out of the atmosphere and turn it into forest biomass. A couple things to think about that are sort of important when we think about reforestation and its relationship to conservation biology is uh, what's the island equilibrium of Central Park, right? There's no space left to reforest. And so this is at probably at its equilibrium limit for species that it can sustain. There's no opportunity for reforestation there. But that's, urbanization is not the only reason we lose forest. A lot of forest is lost to things like the right picture here, where it's due to logging. And so logging creates islands, which you can imagine if this center section hadn't been logged, this is a much much larger area that could sustain many more species. So reforesting this has a much bigger impact, or could potentially have a bigger impact on the number of species that can be maintained here. And all kinds of resource management strategies then go into play with, well, what if we build corridors of forest to link up these habitats? And then you can be smart about the ways that you log, because it's not as though logging is necessarily bad in and of itself, because young forests tend to be very productive forests. Um, but some species depend on older growth. We talked about that back in the population ecology lecture, where some species will actually thrive on the edges here, but some require deep old forest and some require new fast growing forest. So it's a complex issue. The whoops, the globe pictures here, these maps just showing where there's net reforestation versus deforestation. The developed world, right? US, Canada, big chunks of Western Europe and parts of Asia have net reforestation. We're actually growing more forest than we're harvesting. So that is a lot due to resource management practices, where you can cut, when you can cut, how much you can cut, and then how much you have to plant for everything that you do cut down. But then huge swaths of the world, especially developing nations, where there's a net loss of forest. So, like I said, when in the first slide about conservation biology, a lot of this has to do with where do we focus our research and investments in what to protect and how to protect it. So here I just wanted to give you an idea for um, I think this is the most recent year for IUCN Red List, broken down by the proportion of living species. These are named species. That's an important caveat, because there are, for example, in insects, there are lots and lots of unnamed species. Um, as you go, so those little two-letter abbreviations, if you go back to where I introduced the IUCN Red List, right? CR is the critically endangered, endangered, vulnerable is yellow. So if you extinct in the wild is the black. So like here, cycads are the only ones that have an extinct in the wild bar there. Um, but this red, that's an idea, it gives you an idea for how many critically endangered organisms there are. And right, so for example, if you think about mammals, about 5% of the known mammals are critically endangered. If you just go up to endangered, it's something like, 10%, and then going out to 20, maybe 25. The gray area, another thing I mentioned earlier, the data deficient. So I think if the red bars, I should have written this down, are what we think the sort of red, yellow, orange ought to extend to, if we had enough data. But hope is it's not all gloom and doom. For some things like cycads, it's pretty bad. And it's hard to say that anything with, talking about 35 to 40% of the species in some of these groups being vulnerable or worse. Um, it's hard to think of that as not all bad, but if there is any bright side, um, when people first really started paying attention and collecting data for the red list and for really putting an emphasis on what should we be focused on, most of the things that were being studied because somebody already knew there was a problem. Just as an example, I've talked about this a few times. My master's degree, my job, the research assistantship I had was to help Park Service and Fish and Wildlife monitor the reptiles and amphibians on the Space Center and sort of decide what were the important things to focus their attention on to know what was in trouble. And so the things that got the attention were the things that were rare already, or we knew there might be a problem because they need large home ranges to migrate across, for example. And so there are so many roads, we knew lots of them were probably getting killed because we'd find them dead on the roads. All that is to say that over time, more and more species where there wasn't a known problem going in, as those have been studied, the number of species that are doing okay has increased faster than the number of total threatened species. So threatened being the things that are in those red, yellow, orange categories. Now, it's still way too high. Um, right. This is still, 40,000 species. Is that where that? 50,000 species that are threatened, right? That's not good. Um, and so, uh, mainly all that to say, it's not good, but it's not all bad. Trying to look for something positive. If you zoom out in terms of not just thinking about which species or which types of organisms to look at, um, what parts of the world should we be focusing our attention on? Biodiversity hotspots are the ones where it makes the most sense to put the most investment into. These are small areas with exceptional numbers of endemic and tend to be endangered and threatened species. I'll talk about the why that is on the next slide. I mean, it's part of the definition of a hotspot. Um, One of the things that comes up often in discussions of where the biodiversity hotspots are indicator species. And again, to my work at the Space Center, we were looking for which species are representative. Like we can go out and know that we can catch these things, but the rate that we catch them at is going to be indicative of how well sort of the whole community of reptiles and amphibians are doing. Because you can imagine if I picked the rarest thing that is in the most trouble, there'd be lots of days where I went out, or maybe even years where I went out and I could not find one. In my 2.5 years at the Space Center, I found only one eastern diamondback rattlesnake. We knew they were on the site. We knew they were in trouble. But, if I had ended at 2 years, we would think they were extinct on the site. It was not until the third year when I found one dead on the road. Um, and so that's not a good indicator species because you catch them so rarely that they're not you don't catch them enough to indicate what's going on. So you want to find something that is a little more common that you can sort of count on catching lots of, but in years where you can't catch very many, that tells you something about what's going on in the environment. So uh, indicator species can be important, especially in these biodiversity hotspots, where you want to be able to go in and get a good assessment of how that ecosystem or that community is doing in any given slice of time. So just a little bit more about biodiversity hotspots with a few examples. Like I said, the two key criteria are that it has high endemism. That is, it's got 1,500 species of vascular plants, and it's defined on vascular plants just because those tend to be, especially in most of these hotspots, the indicator species. I suppose that's a good way to put it. Where at least 1,500 of them are endemic, and endemic means they're just native to that region. And then the second thing is that there's been at least 70% of the original native vegetation is lost. Okay, so they're heavily threatened. So that's what I meant on the previous slide about they're sort of defined by being in trouble. It's what makes them a hotspot. Not just the fact that they have lots of species. So the key feature here of these, whoops, oh, come on, habitats is that even though they're shrinking, only 2.5% of the Earth's land surface, they hold more than 50% of the world's endemic plant species. And it goes along, it's a huge, I don't know the percentage, but it's a huge number of the animal species as well. Um, it's less than 50%, but still disproportionately high given the amount of land surface that they occupy. And so that makes them a priority for conservation. If you think about where can I do the most good in terms of conservation, go to the place that has the most diversity in the smallest area and see what you can do in terms of resource management and corridors and habitat restoration. Uh, what can you do? These examples are ones that get lots of the attention. Just in case you're, uh, they correspond with this map as well, in case you don't know where some of those places are. Um, Different organizations have slightly tighter or looser definitions for what they want to, not what they want, what they define as a hotspot. Um, this is the most general. Some of them are more strict. I think there are 34 in the organization where I got this map from, but it varies from one organization to the next a little bit. They all, in principle, have this definition: small area with lots of endemic species. Okay, that's it for the conservation biology lecture. Again, sorry I couldn't be there in person. I will post the little breakdown of how many questions come from which unit. I'll probably do that on Friday, um, if not sooner. Uh, and so that will give you an idea how to focus your attention, your study time for exam 3. And then I'll also do the same thing for the final. But again, the best ways, just like the previous exam, the best ways to study are the quizzes. Go back and look at your quizzes, take the practice exam, make sure that you understand those questions. Um, yeah, and I will see you Tuesday. Happy studying.